Hi everyone! What I'm going to do today in this short video is kind of look at the structures of the sarcomere, and we're going to kind of review these structures and review this kind of little lesson that we did in our face-to-face class. So, what is the sarcomere? The sarcomere is the fundamental contractile unit of the muscle cell, and it's going to have proteins such as actin and myosin, which are going to interact with each other to shorten the muscle cell. So what I'm going to do here is I'm actually going to draw two sarcomeres side by side just to be able to illustrate some of the striations that we see when we look at the muscle under a light microscope. So the border of the sarcomere is going to be bordered by this kind of zigzag shaped proteins called the Z lines. That's nice, zigzag Z lines. And so what we can see here is we actually have two sarcomeres, one located here and another located here. In the middle of the Z lines, we're going to have a protein, we're going to have a set of protein fibers that are going to create the M lines. M lines, think M for middle. They're going to be right in the middle of the sarcomere. So these structures are going to help attach to the contractile proteins that are found in the sarcomere and help stabilize them into position. On the M line, we're going to have the myosin attached. And remember, myosin is going to have kind of like a golf club shape, and like many myosin will be attached to each M line. So it should look something like so. So in blue, we have the myosin. So to remember that myosin attaches to the M line, remember the little saying M&Ms. So they both start with M, so they're going to be attached to each other. On the Z line, what we're going to have instead is we're going to have the actin molecule attached to the actin molecule attached. So we have that over here, and this is gonna, when we have lots of actin molecules bonding together, they actually look like they are a helical shape. So we're going to have something that looks like this for actin. And again, remember the saying A to Z, actin to Z lines, so to remember what is attached to what. Remember, actin actually has some regulatory molecules attached to it. So in addition to having that molecule attached to the Z line, we're going to have two regulatory molecules. One regulatory molecule is going to be this tropomyosin. The tropomyosin is going to bind to the binding site where myosin would normally attach. So the myosin and the tropomyosin are going to compete with each other for the actin binding site. There's another regulatory molecule that's going to be found on tropomyosin. This is called troponin. Remember, troponin will actually bind to the calcium when that calcium is released. And troponin is going to be responsible for moving tropomyosin out of the way so that myosin has the ability to bind to the binding sites. So this is going to be some of the structures that we're going to have. So just to review these structures, we have the Z line, the M line, myosin, actin in red, tropomyosin over here in green, and then troponin located here. We're going to have some bands that are going to form, so let's talk about these bands. So if we go from myosin head to myosin head, this area is going to have an area where actin and myosin are going to overlap, and because they overlap, they create a shadow when light passes through. So they're going to be, because it's going to be so dense. And so this area, we call it the A band right here. And so we would have an A band located here and an A band located here for the second sarcomere. However, there's going to be an area from this location of the myosin head and this location of the myosin head. It will be actually composed of sections of 2 sarcomeres. This area from here to here is going to be the I band. And it's only going to have like actin. There won't be myosin present. The I band, when we look at it under the microscope, is going to look lighter because that area isn't going to be as dense. And so this darker area and this lighter area is going to be what's causing the striations when we see muscle under the microscope. I always remember A is dark because there is the letter A in the word dark, and then I is going to be light because there's I in the word light. So that's how it can help me keep track of these. There's another little kind of band or zone. This is going to be encased in the A band. It's going to be an area where there is kind of no actin located here, and so we call this the H zone or the H band. I think of it as a hollow zone that is present in this area. And then again, this whole place over here, from this area is going to be the sarcomere. So I'm going to put S there for now. When the muscle contracts, these bands will actually change length. This is part of the sliding filament theory. And to remember which bands change length, I always remember the word shrink. In the word shrink, there is an S for sarcomere, so that will get shorter. There is an H for the H zone and an I for the I band. So these 3 structures are going to shorten when the muscle contracts. The A band is actually responsible for pulling on actin, and so the myosin is responsible for pulling on actin. So the myosin is not going to change length. So the A band won't change length. So there's no A in the word shrink. So I remember that doesn't change length. So the sarcomere, the H zone, and the I band are going to shrink. The A band is not going to change length, and no structure is going to get longer during contraction. So I hope that helps for a bit of a review. I did post a kind of blank version of this drawing so that you can actually practice it. And I will see you in the next video. Have a great day. Bye, my little nerdlings.